Welcome to the Maine Science Podcast. I'm Kate Dickerson. Today's episode is a conversation I had with Caitlin Howell, an associate professor of biomedical engineering at the University of Maine. I have been a big fan of Caitlin's for a long time, and getting to talk to her about her work only made me a bigger fan. Caitlin's focus on looking to nature for inspiration, using a truly interdisciplinary approach to problem solving and solutions, and her interest in having her work get out of the lab to the rest of us is a source of never-ending inspiration. And we didn't even get a chance to talk about her Maine-focused work, which you can learn about from her 5-Minute Genius talk at the 2023 Maine Science Festival. I've included a link to that in the show notes. I hope you enjoy this conversation as much as I did. So, Caitlin, welcome to the Maine Science Podcast. It is a delight to have you here. I've been a fan of yours for a very long time, from afar. I'm not even sure if you knew that. Before we dive into what I think is your incredibly cool and interesting work, I was hoping you could give us a bit of a background about where you grew up, how you got into science. Like, when was it that you knew that science was something that you wanted to do? And just to make it clear, I know you're a bioengineer and you do a lot of engineering. I consider engineering and science, from my perspective, to be two parallel pieces that are vitally important, and they both need each other. So my definition of science is very broad.
I totally agree. And I think my story is really going to illustrate exactly why you are correct in that. So I'm actually a Mainer. I was born in Lewiston and actually grew up in the western part of the state, Oxford Hills, one of the very tiny towns. Small town Maine. Love it. Really important, really formative. I knew from the time I was a little girl that I wanted to be a scientist, but I thought I actually wanted to be a marine biologist. And so I spent a lot of my early years memorizing the scientific names of whales because I was pretty sure that that's what marine biologists did. So I ended up going to study biology at the University of Maine. And while I was here, I tried to find a job in marine biology, but unfortunately, there weren't any marine biology labs that were accepting first-year undergraduate students. So I couldn't do anything there, but I got really lucky and I happened to overhear my mycology professor trying to recruit another student near me for work in her lab. He had no interest because mycology, as some of your listeners might know, is the study of mushrooms, fungi. And that's not nearly as cool as whales, right? Wrong, actually. She accepted me into her lab and I started out doing dishes, but I just sort of started learning all of this amazing stuff. I think we don't really get to appreciate just exactly how amazing they are and what they do for us. But when you're face to face with them, it's really very clear to see. And I started to get really, really excited about these things and really want to understand just exactly how are they the organisms that are the great recyclers of our planet? How do they function? How do they do what they do? It's still way understudied in my perspective for what it does for us. So that was really my first taste of this is actual research. It's not, memorizing scientific names. It's actually asking the question of, well, what is happening here and why, and digging into it. And I fell completely head over heels for it and have done nothing but that ever since.
So before I let you go into the rest of how you ended up back at Maine, why do you think marine science from the landlocked world of Oxford Hills? I mean, I remember I had a conversation with Deb Bronk, and she grew up in the Midwest, and she said kind of the same thing. At 6 years old, she was collecting seashells from anywhere she could get them. And it was usually yard sales because it was Midwest. Like, she just had this affinity. I'm just curious, was it the whales? Because they are pretty awesome.
Whales are really cool, but I think actually the landlocked part of it is a little bit of that. I knew that I wanted to be near the ocean, doing something near the ocean. That was definitely for me. So I guess it's hard to say though when you're 6, right? Maybe I saw a television show or heard a song or something and that just stuck.
So after you found out about the remarkable world of mushrooms and fungi, which I also find really fascinating and a little intimidating, I think they are vastly more important to our ecosystem than we give them credit for, and at times dangerous. And I don't mean that as a flippant thing. I think we really do not understand the impact that they have and can have on us. After you get through with that, what was next?
So after that, I actually stayed here at the University of Maine to do my master's degree. I had told my advisor that I wanted to do more in engineering and the materials. So fungi, when they grow inside a piece of wood, for example, they are physically changing it and they are becoming part of the material. And that was really interesting to me. And I wanted to study that from an engineering perspective. And she was amazing. And she said, yes, but I can't give you a degree in engineering. So you can do this master's degree in biology, but I'll let you go play with the engineers. And she did. And so I actually got to play with some of the really, really big instruments that shoot X-rays at stuff and start to figure out exactly what's happening with the cellulose crystals. And it was wonderful. But at the time, I got approached by one of the chemical engineering professors, and he said, hey, you've been over here playing, and that's really cool. We have an opportunity to send a student to Germany to study with a group over there for 5 months. We usually send chemical engineers, but we don't have any at the moment. You want to go? And if the answer is yes, you have to leave in 2 weeks.
Wow, no pressure there, huh?
But it's one of those moments in life when you're like, okay, total change of direction, let's go. And when is the time to do that? When you're, early in college. So I did it. I dropped my entire life, again, with the blessing of my absolutely amazing advisor. She let me pause this, and I went over and I lived in Germany for 5 months. And there I got to use even bigger machines. I got to use class 4 lasers to shoot at materials and see what was coming out. And I tried to do it with wood and fungi, but unfortunately, it just burned my wood and fungi. So we backed it up a little bit, and we were just looking at molecules on surfaces instead. And that was really cool. We got 2 research papers out of that 5 months. And so the professor there in Germany came up to me and said, hey, you want to come over here and do a PhD? So I'm like, yeah, just give me a minute to finish my master's degree. I'll be right back. And so I did. I came back, finished my master's degree, and moved to Germany. And that group was actually studying physical chemistry, which for those listeners who don't know is studying sort of the physics side of chemistry. So it was very far away from biology. But at the same level, we're all trying to do the same thing, understand what's happening. And so I got to bring a flavor of biology to this group, which was across the board physicists and chemists. And that was really my first taste at truly, truly interdisciplinary research. And we found that working together, we were able to make some really incredible and powerful discoveries. I think I ended up with 11 publications out of my PhD, just because it was such a fruitful area. There are so many interesting questions at this intersection of disciplines that we're really starting to get the hang of answering.
So for people who don't know kind of the inside baseball for academics, 11 papers out of a PhD is extraordinary. Usually I hear maybe 3 in a good thesis. Like that is an extraordinary amount. That's just a little informational tidbit for people who are listening. I'm curious what the culture shock, if any, was. I mean, did you know German? Did you have to learn it? Were you able to do stuff entirely in English? Like that is a non-trivial change of plans to go, not just switching into a truly interdisciplinary, outside your field PhD, but doing it in a different country with a different culture.
Yeah, I had no idea what I was getting into. I could count to 3 in German. That was pretty much it. But yeah, it was a culture shock, but a good one. And, any of your listeners that are in the position to go and do some work abroad like that, really embed yourself in the culture more than just a vacation, I highly, highly recommend it, because I think there are a few things in this life that will stretch you quite as much as living in a different country. I learned that the way that it works is we are very lucky as native English speakers because English is the language of science. And that means that things are just naturally a lot easier for us, writing about our work, presenting about our work. We don't have that additional barrier. And in fact, it was possible. There were students in that lab who spoke very little German because they could get by and do everything that they needed in the science world in English. Now, how did they do stuff at the supermarkets or restaurants? I have no idea. Somehow they made it work, but that was possible. But while I was there, my PhD supervisor actually offered to all of his international students, if they wanted to take German lessons, he would pay for them. And so I absolutely took him up on that, and I became fluent in German. And I could finally talk with the technicians in German, which you get so much more information when you talk to people in their native language. At no point was I very good, but it was passable. So that ended up working out. But I actually found that I was able to help a lot of my colleagues with writing and things like that because of the English language.
It's so interesting because then that lab was not only vital for interdisciplinary work, which I think has been kind of the secret sauce that people don't totally realize is really critical for advancing science. Yeah. but also from the perspective of this merger of cultures and having to truly find a common language where you can all communicate effectively. It sounds like it's this little microcosm of exactly how best to go forward to learn things.
It absolutely is. And I really love this age of technology where we can talk to each other across continents, at the speed of light. It's fabulous because we really have a lot to learn from each other. So while I was in Germany, I learned that they approach science in a different way that I hadn't realized. They, at least in that group that I was in, were very, very focused on the basic research. And if you gave them a few beers, you could actually get out of them that, the Americans, you're always really focused on applications. How is this useful to people? What are you going to do with it? But we need to understand the basics, right? We really need to go to the fundamentals, and who cares how it's used? We just want to understand it. And I think it's all important, but it was really eye-opening to see that reflection of research in the US from a different perspective.
I wonder how much of that is culturally, we are such a younger country and have experienced as a society so much less than anywhere in Europe that they just come out of it with a different perspective. I'm with you 100% that we were lucky enough to go to Ireland for a year when my kids were little. And even though they didn't have to learn a new language, it was still a different culture. And I really, truly believe that changed their lives for the better and gave them an understanding and an empathy in ways that they wouldn't have had we gone even down to Boston for a year or something. It changed how they looked at the world.
Absolutely. I bet you're right. What a fantastic experience for them.
It was pretty great. So then I know you went to Harvard for a postdoc. If you don't mind, and I'm asking you to do this in particular because you're so good at communicating this idea, could you explain why people do postdocs and why it almost becomes a necessary part of research science in a lot of ways? Because I think a lot of people think, well, you got a PhD, what could you possibly do after that? Like, what do you mean you have to have another temporary position? So if you could explain just a smidge about a postdoc, but then also what you did at Harvard, that would be great.
Yes, absolutely. Happy to. So when you start your PhD, you go to a group of a person who is doing research, and this person helps to train you to be able to do research like they do, right? This is the field that I work in. This is how I do the things that I do. But really what we need in science is people who can come up with their own ideas. Hey, nobody's looking at this. We really need to start to dig into this and understand that. And what happens if we combine these things? It's not impossible to do that if you've only done a PhD in one group, but it becomes harder. So people do postdocs to sample another way of thinking about things, another way of doing things, another field of research. And then they have multiple buckets that they can pull from, multiple fields that they understand to be able to say, hey, everybody, there's a big gap right there that nobody's looking into. I'm going to step up, I'm going to have a research lab, and I'm going to study that. Doing a postdoc is what allows you to do that. And so when I finished my PhD, I was still really enamored of microbiology, right? Getting back to the fungus and the materials. And I really wanted to look into how we can control biological systems with materials. And, maybe even how we can get materials to grow or have a part that's growing. So I realized that if I wanted to do that for research, I needed to have some materials science chops, right? Some of your listeners might not be familiar with materials science as a field, but basically look around you. There is stuff all around you. Someone has had to think really, really deeply about how strong that stuff is, how wear resistant that stuff is, how flexible it is. All the properties of the things that we use every day are really, really important. And so there are scientists that study that, understand what we have, and make new materials. So I started searching the internet for somebody who was doing some combination of microbiology and materials, and Harvard popped up. The Wyss Institute for Biologically Inspired Engineering is doing some absolutely incredible work. And so usually when you're in academia, you apply for a lot of things and you don't get most of them. It's like job searching anywhere. So I was fully expecting no answer, crickets, or a flat out rejection when I applied. But actually, they wrote back to me and said, yeah, we got some open positions. You want to come on over? So that was amazing.
I have a feeling that those 11 publications during your graduate work certainly helped them realize they were dealing with someone who knew how to do some kind of research, and that probably didn't hurt any.
Probably not, but I'm still very, very grateful because that's a hard place.
So how long were you at Harvard?
I was at Harvard for 3 years.
So a fairly longish postdoc. I know some fields you could do 3 or 4 postdocs of 3 to 5 years each before you find a permanent position. So 3 years is manageable, it seems to me, and an opportunity to figure out where you might want to slot in.
Exactly, yes. And it does differ across the disciplines too. This particular postdoc was interesting because the Wyss Institute for Biologically Inspired Engineering does more than just sort of the standard science that you might think of. They do a lot of technology translation, which is, hey, we have this thing, there's potential for it to help people. Now let's do the work to actually get it into the market where it can help people. There's a pretty significant gap right now between building something or finding something in a lab and getting it to the point where it can actually be useful. Because it takes a lot of money, it takes a lot of time, it takes a willingness to go out there and interview people and be a business person. And a lot of people who are science-minded, that's not their favorite thing to do. So this institute was created in part to really start to fill that gap. And so I got an opportunity not just to study materials and microbiology and where they intersect, but also really get familiar with the process of technology translation. How do we think about things from a commercial perspective? What sorts of steps are needed for this? How can we start planning from the very beginning to be able to get something out into the market? So that's another reason why it was longer.
So that's really interesting because it's another kind of interdisciplinary approach, but it also harkens back to what you said about the difference between culturally basic research versus applied. That is a super applied approach. The basic research has already been done. So you really took a jump back into American culture with that.
Yes. Welcome back to America.
No, I mean, I think it's really cool and fascinating. I think one of the things that causes misunderstanding in how science works is that there'll be an announcement of some really great thing that scientists have figured out, but they don't know how to scale it up because that's not their bailiwick. And so then people are like, what happened to that thing we heard about 5 years ago, which leads to books like, Where's My Jetpack? Right? Like,
Absolutely.
Okay, now we're at the cool part of your UMaine work, which is when I became a fangirl. So Inspired by Nature, which we have actually run a session at the festival called that because it's just so cool when you think about that nature has figured out things over millions of years on how to work and why reinvent the wheel. So hit it. Tell us, you've got a full open mic. Go ahead and do what you want and tell us in whatever detail you want, in all seriousness, like what it is you're looking at. I imagine that you've got different projects, I assume with different students and different foci. So if you want to kind of touch on all of them, that would be great. If you want to hit a few, I will periodically jump in with questions based on what I may or may not remember on what you've been working on.
Yeah, no problem. Got here, started a lab, and the lovely thing about research is that you get to be like, okay, I know where I think the problems are, so I'm going to go after them, right? It's the best job ever. So we have now several projects where we're really on a 30,000 foot view looking to understand how biological systems interact with materials, and then use that understanding to control those interactions. So the projects that I'm working on are great examples of this. I'll start with the first one, infections. We all know them. We know that they're a big problem. We know that they are getting worse with the rise of antimicrobial resistance. I mean, this stuff is really, really scary. Nobody wants to go back to the world where a cut finger could mean death, right? So we are working on ways to understand how materials can play a role in stopping that process of infection. So materials are a big problem in this because usually when you end up getting infections in humans, it's something is put in your body in some way, and that can bring microorganisms with it that can stick on that surface and then begin to proliferate. That's not the only way that infections start. You can do it with just bacteria, but materials make it a lot easier for bacteria to grow. So in this project, we're focusing specifically on catheter-associated urinary tract infections. So catheters, for those of you who don't know, who are listening, are the most commonly used medical device, and they allow urine to drain from the bladder. So anybody who is going for surgery, who's going to be out, will need this type of thing. Women giving birth would need this type of thing. People who have lost the ability to control the muscles would need this type of thing. They're really common, but just because of the nature of where they are, come with a lot of bacteria and a lot of infections. And this leads to so much pain and misery. It's just terrible. So right now, the treatments are you give the people antibiotics. And as we're learning more and more about our microbiome, our bacteria, we know that not only, are we contributing to resistance by doing that, but we're also probably damaging the natural way that our bodies are keeping everything in balance. So we were looking at ways of being able to not use antibiotics and to stop bacteria. And as you mentioned, we do bio-inspired engineering, another thing I picked up at the Wyss Institute. And so, nature, millions of years, has figured out ways to solve problems. Well, let's look at nature for a solution to this problem. So if you want to think about the most relevant example of how nature controls bacteria, look at us. We're superorganisms. We have so many bacteria in us, and they really contribute to who we are on a fundamental level. But our bodies aren't using a lot of antimicrobials, antibacterial compounds to keep them in check. It's doing other things. So we said, okay, let's look at the place in the body where there are the most bacteria and start to just pick apart a little bit how those bacteria are kept in check. And that would be our intestines, right? Tons of bacteria in there. And if we think about how our bodies keep our intestinal bacteria happy, they use a lot of tricks, but I think the fundamental component is really mucus. So most of us might hear that word and think, ew, that's gross. But I would love everybody to take a minute and just really appreciate how amazing mucus is as a material. So what it's actually doing is it is creating a physical barrier that is stopping the bacteria from getting down into the intestinal cells. It just stops them. But it doesn't stop them like a solid surface. It's actually liquidy. We all know it's moving, it's flowing. But as the bacteria start to try and tunnel into it, it gets thicker and thicker, and then they can't. But because it's not a solid surface, they don't go, oh, here's a nice place for me to stick and grow. They just go, hey, I'm kind of happy here. I'll just chill out. And so that component of just having a mobile liquid barrier was something we said, hey, I wonder if we could copy that. And I wonder if we copied that, what sort of effect it would have on bacteria on these surfaces. So it turns out we could actually create a mucus mimicking layer on catheters, urinary catheters, really, really easily. All we were doing was taking a biocompatible liquid, doesn't mix with water. We can infuse it into the materials that catheters are already made out of. So we're using the same things that are present in hospitals. And when we do that process, we end up with a liquid-like barrier system. So we tested this out in the lab, and it worked surprisingly well. When the bacteria weren't seeing a hard, solid surface, they weren't sticking and growing and creating compounds and spreading like they were when the material was solid. So that was really, really exciting. But in materials research for medical applications, if anybody ever tells you, hey, this works great in my lab, never believe that it's going to work great in a real world situation. Because we are so beautifully complicated. There is so much going on in our bodies that we could never replicate that in a lab. So we actually made some science friends. We got some collaborators at the University of Notre Dame who are experts at catheter-associated urinary tract infections and really understanding the biology behind it. And we're like, hey, you do this type of stuff in living systems. We want to test some of our samples and see how it works. This then is an example of why basic research is really, really critical, and why on some level the Germans are right when they say we need to get the basics, right? Because my collaborator there, she said to me, yeah, but in our research, we've been finding that it's not actually the bacteria sticking that is the problem. The problem is actually proteins sticking. We probably know that whenever you take a foreign object and you put it into a body, the first thing that the body does is it tries to wall it off, right? It tries to protect it. So the way that happens is through a protein called fibrinogen. This protein is absolutely amazing. It's what stops your blood flowing and gives you a clot. It's also what your tissues produce to try and make stuff go away. Turns out that when you put a catheter into a bladder, the same thing happens. This catheter is bouncing around. Even if you're lying in a bed, stuff is moving constantly, bouncing around. The cells, the body is saying, oh, foreign object, release the fibrinogen, wall that off. And it does. Tons of fibrinogen comes, and it's a very beautifully sticky protein. It sticks to this catheter. Unfortunately for us, bacteria have evolved over millions of years to recognize that the presence of that protein means that there's probably a pretty good place for them to be growing. Maybe there's a wound, maybe there's some other opportunity. So they have apparently evolved to be able to recognize this protein and stick to it. So my collaborator said, would your system work against protein? Because if we could maybe just not let the fibrinogen stick, this might really help. And so we said, sure. I mean, mucus also stops proteins from sticking. So why wouldn't it work with this? So we sent her a bunch of samples and she tested them in her model, living system. And the results were absolutely jaw-dropping. We saw between a 90 and 100% reduction in the amount of bacteria that were sticking, massive drops in the amount of proteins that were sticking, all without antibiotics. Simply by mimicking just one small part of how nature does it, we were able to get this astounding result.
So are you surprised that your target, what you were designing was mucus? You didn't even think about the protein and it still worked?
Yeah. And this is why it's really, really important that disciplines talk to each other and different researchers talk to each other. So I would never have dreamed that that was important. But thanks to her research and her insights into the basic biology, combined with our material science, we were able to achieve something that we hadn't seen before.
That's really amazing. I have one other question, and this might be too weedy, but you said you developed it so it's integrated into the material itself.
Mm-hmm.
So does it feel the same on a macro level to the people who are using it? So the people who are inserting it, like they don't notice any difference in it. It's just atomic level, so to speak, or the basic building blocks of the material are different. Is that how it works?
That's a great question. If you take your finger on it and you slide it, you might notice that it's a little bit more slippery. But yeah, it's fundamentally integrated into the material.
I would imagine little slippery might actually be better for a catheter.
Yes. And also, we had to do a lot of talking with doctors and nurses and healthcare workers who use these things to really understand, because again, we want to translate this. We want to get this out there to help people. So we're understanding from the very beginning what the process is to make sure that our system is compatible with that process. When they apply these things, they will frequently not touch the surface at all, because if you touch the surface in traditional catheters, you get bacteria on them, you insert that into a person, that's not good. So the process for many of the current commercial options is a no-touch process. You're touching the catheter, but through a protective layer so that what you put in is sterile, right? It doesn't have any bacteria on it.
Super interesting. All right, give us another project. And by the way, when you said jaw-dropping, after all that whole lead-in, I thought you were going to say it was a total failure. So that's actually even better that it wasn't.
I was once told if 80% of your experiments don't fail, you're not asking interesting enough questions.
That's a harsh marker, but I understand it.
And I have so many failures. Think about that all the time. So, okay, yes, that was really exciting. But another thing that we're doing in this lab is taking the next step. So in that, we're trying to get rid of the bacteria, right? No bacteria, no bacteria. We don't want anything sticking, which is okay for that application. But I really, really think that our human thinking of we need to get rid of all the bacteria is not really a sustainable one, even if we end up doing it in ways without antibiotics. I really feel that bacteria are a critical part of our world. They're a critical part of us. They're around us everywhere. And if you really think about it, they're super cool. I mean, these teeny tiny little machines that can do stuff and make stuff. And it's incredible. So I wanted to explore a bit more of how can we stop fighting against these organisms quite so much? How can we start working with them? So you might be familiar with some of the microbiome research that's coming out just daily, new recognition on how important it is, how stuff interacts, what it affects, everything. And so there's really a lot of this research going on. So I really thought that we could harness this growing body of fundamental research and figure out how we can start driving systems in the direction that we want them to go using materials. So another bio-inspired example, if you think about your materials, your hands, your skin, your everything, it also is not a static material, right? It's always changing. It's constantly growing. It's sloughing off. It's adapting to the environment. That type of material, a changeable, living, dynamic material, is really, really powerful in a lot of ways. So how does that happen? What is it about our materials that make them be able to adapt and change? And then can we apply that to a maybe not living material to get it to adapt and change to a living component? So we're complicated. There are a lot of things going on. But if you walk it down to the very fundamentals, I would argue that the thing that makes it all possible is our vascular system. So our vascular system, our veins and arteries and our blood, is what allows our tissues to be able to continuously do stuff. It brings energy and oxygen. It takes away waste. And in doing this, it also moves signals around. All of our tissues are constantly communicating through our vascular network. So we asked, can we replicate that? And it turns out we can. We actually can 3D print empty vascular networks into materials. And interestingly enough, we can make it into the same types of materials that urinary catheters are made out of, and a whole bunch of other medical devices are made out of, silicone rubber. And so we built a bunch of these things, and we asked ourselves, can we use this vascular network to control bacteria on surfaces? So it turns out we could. We could actually inject into our vascular network a compound that would direct the bacteria to do something. So die or grow or stay static, right? There are chemical signals that we understand will tell bacteria to do this. And so we can put those materials into our vascular network. The materials will diffuse up to where the bacteria are growing on the top and will affect those cells. But when we started to play around with it, we actually found that we could do even more than just this. So we could listen to the bacteria. At the same time that we are pushing materials up to the bacteria to tell them to do stuff, those bacteria are actively growing and changing, metabolizing, creating compounds, some of which diffuse back down into the vascular network. We can then collect the liquid that's in the vascular network and test it. And we can see how far along the bacteria are in their growth process, how many are there, what species they are. So we're super excited about this because we're envisioning a new type of material system where it's no longer just it does nothing. And then when something bad happens, we have to react to it. We can create a material where we can have a continuous conversation with these microbes and move to an area where we're working together and we just sort of nudge them in the direction that we want them to go continuously, and we can adjust, like a living system. So that's another project I'm very excited about here.
That sounds so much to me like science fiction in all the best ways, but it leads me to think like there is a certain audacity into saying, well, let's just create a vascular system for these materials. And a good audacity, right? But that is a lot. That is a super big reach. And I'm going to assume, and you can correct me if I'm wrong, that part of the reason you're able to do these two super innovative, really interesting projects is because you're not doing it by yourself and you've got a research team. First of all, how big are your teams working on this? Is there any overlap? And are you trying to model in your research lab what you've learned works when you did your PhD and your postdoc, which is interdisciplinary approach?
Yes. So first of all, you are absolutely right. I get to be the one to stand here and talk to you. But the thing about being a principal investigator, we say in a lab, is that we're really the figurehead and the students are the one that make it go around. So I have 5 really amazing graduate students right now. And then we have a team of undergrads too, who come in and out as their schedule allows. And they're working together on these projects. And they're really the driving force. So I teach them how to do stuff. I set them on a direction, but then they are the ones that really start to go, oh, and then I read this type of thing. So let's try this, or that isn't working. So we're going to do it this way instead. And so a lot of the credit goes to them for being able to do this work. Graduate students are what make the research world go around, 100%. But yes, we're very much working at the interface of disciplines when we do this. And we have a lot of amazing collaborators who help us. We're not a research island. We can't be. There is so much stuff you need to be able to do. You need to have experts in these different areas who can work with you and help you out. And that's the only reason why we're able to really be expanding the way that we are at the moment.
Do you think it's helpful that you have graduate students who have an advisor who understands the value and the importance of reaching big, but they also don't necessarily carry with them years and years of frustration and failure? So it doesn't occur to them that building a vascular system may be harder than they think.
Yes, no, you're actually right. Like the guy in the anecdotal story where a guy came late to a math class and there was a problem written on the board and he thought it was homework. So he took it and solved it and brought it back. And it was one of those problems that had been a mystery in the math world forever.
I love that.
Yeah, we definitely got to do more of that. But exactly, you just say, hey, could you do this? And they, for the most part, will find a way to do it. And it's very incredible. But also, I see my role as really much being the umbrella and getting all of the stuff out of the way so that they can focus on nothing but research and being creative and that type of thing. And I think for that reason, anyone who's listening, who's considering going to graduate school, that can be one of the absolute best times of your life where you're not just like, okay, I'm going to contribute to the sum total knowledge of humanity. I don't have to worry about paperwork. Someone else is going to pay me. I just have to show up in the lab and ask questions and find stuff.
So do you get as much inspiration from your students as it seems to me that you do? Like, it seems to me that you feed off of their asking, what if, why not? Is that fair?
Yes, absolutely. Nothing makes me more excited than being able to sit down with them and just consider what it is that they found and what they're thinking about. And I got to tell you, Kate, there's something really, really important about students that people who are not in the sciences might not realize. But students are really critical to our enterprise for another reason, and that's that they think outside the box. They have incredible fluid intelligence, right? You give them a problem and they will approach it from ways those of us who have more crystalline intelligence, who have been working this for a long time, would never even think of because it's outside of our box. And so that's why it's so important to have grad students, but also undergraduates who are very, very new to research, to be able to provide that perspective. And it just takes that one insight of, hey, I thought I heard about this random thing over here that's unrelated. Could this be something that's happening? So it's really important. It happens all the time. And when it does, it is so exciting. There's nothing better.
I love that you get to have aha moments generated by your students on what sounds like it could almost be a daily basis. I mean, what a win. Mm-hmm. And I know you just said that being a grad student might be the best job of all, the best time of your life. But I'm thinking, Caitlin, that you have slotted into a position here as a faculty member where you get to experience a lot of that joy in little microdoses to keep you going.
Absolutely. And I'm just really grateful that we live in a society where we can do this type of thing. Another thing that I realized when I was in Germany is that our research enterprise here in the United States is really incredible. We have to compete really hard for grants. I always wish there was more money for science. But the reality is that we do pretty well. We are answering the questions that need to be answered because we have government agencies like the National Science Foundation, the National Institutes of Health, the Department of Energy and Defense, and all of this stuff that do care about science and do fund those top projects that are likely to make a difference for everyone. And so I really have a sense of gratitude about that. We're doing well.
I can't think of a better way to end our conversation, with the only note being, honestly, we could keep going for hours and hours, but I did not ask for that much of your time, so I won't take it. I love hearing about your work. We didn't even touch on some of the Maine specific stuff, so I'm going to point people to your 5-Minute Genius talk and figure out a way to have you come back at some point so you can talk about Maine specific stuff, because I find that equally important and energizing as part of my continuing pushing the boulder up the hill of reminding people that Maine scientists really are all that, which I think this conversation has proven, but there's aspects of Maine itself that are really inspirational. So that's my little plug for you, and I are going to continue this conversation at some point.
Sounds good. Thanks so much for this opportunity, Kate. I really appreciate it.
You are very welcome. Thank you.
Thanks for listening to the Maine Science Podcast. Make sure you never miss an episode by subscribing on your podcast platform of choice. And please leave a rating and review. It will help more people find us and help spread the word about some of the remarkable people doing science in Maine. The Maine Science Podcast is recorded at Discovery Studios at the Maine Discovery Museum in Bangor, Maine. The Maine Science Podcast is executive produced and hosted by me, Kate Dickerson, and edited and produced by Scott Loiselle. The Discover Maine theme was composed and performed by Nick Parker.